Hi everyone, my name is Seon Chitrananda and I'm super passionate about how we can use machine learning to fundamentally understand human genome biology. So 40,000 years ago, we domesticated wolves for the first time. And this is a huge inflection point for humanity because these wolves allowed us to defend our food from other animals and survive. And through the process of evolution, these wolves have now become the dogs and puppies that many of you own today. So through these random mutations, humans have been able to adapt and survive, but they've also become our downfall. Because the thing is, these random mutations cause genetic disease. Over 6 to 8% of the world has some form of a genetic disease. That's 400 million people across the globe. And so over the past year, I've been focusing on developing machine learning methods to fundamentally predict DNA protein binding and detect genetic diseases in our genome. So because biology is so complex, the process for building therapeutics for genetic disease is also inherently complex, time-consuming, and expensive. On average today, it takes over $2 billion to develop a drug, and 12 years. And so the 3 billion A's, C's, G's, and T's that code for life are inherently complex. So how can we really try to solve this problem and convert the genome into actionable information? One positive trend that may be changing this is the rapid increase in genomic data. Modern genetics is experiencing a data revolution. Every year, we're seeing a 2.2x increase in the number of open source genomic data sets. And this largely can be attributed to the first human sequence. We sequenced the first human genome in 2003, after billions of dollars in investments and 13 years of research. But back then, it cost $100 million to sequence a single human genome. But through the work of revolutionary companies like Illumina, we've been able to sequence a human genome for only $47 today. For the cost of 2 pizzas, you can now unlock the genetic code that codes for life. Just think about that for a second. And so currently, we can not only sequence our genome, but edit our genome using a gene editing mechanism called CRISPR-Cas9. This technology allows us to potentially edit out any single mutation in the entire genome. But our ability to leverage these technologies is severely impaired by our lack of understanding of the genome. When you think about the genome, it's essentially a book of A's, C's, G's, and T's. And if you thought reading Shakespeare was tough, trust me, reading the genome is far more tougher. So our ability to really convert the genome into actionable information in order for us to predict genetic disease and fundamentally understand different processes such as DNA protein binding is really hard. So let's take a step back and try to understand how biology works on a really high level. We know that DNA undergoes transcription to form RNA, and RNA undergoes translation to form proteins. These proteins are made up of thousands of amino acids, all of which are encoded in our DNA. The DNA is essentially the source code to which proteins are the output. A really good analogy that I like to think of is that the protein is the conductor to which the DNA is the sheet music. The protein carries out many of life's necessary functions for survival based off the instructions of the DNA, or the sheet music. So when we think about how mutations in DNA alter our body, they essentially fundamentally alter the structure of these proteins. So as a result of mutations in our DNA, we may either have dysfunctional proteins or simply not enough proteins that are produced, because mutations in the DNA may make it less attractive for the process of transcription to occur. So without enough of these proteins, many functions in our body just simply don't occur. And a really good example of this is Rett syndrome. Rett syndrome is a rare neurological disease that occurs in 1 of every 15,000 girls across the world. And it can be caused by as little as 1 single mutation in the MECP2 gene. And as a result of it, children with this disease have severe cognitive impairment and their nerve function and severe damage to nerve function. So it's likely that children with this disease may never walk, they may never talk, they may never eat without assistance. And so with the rise in genomic data, more powerful computing, this is no longer becoming a biological problem. This is now a machine learning problem. There are so many experiments and facets of genomics research today that simply involve recognizing patterns in iterative experiments. And all of you in this room know just how powerful machine learning is at recognizing patterns in the genome. And so today I'm going to be talking about how we can use convolutional neural networks to predict how likely a transcription factor protein will bind to a DNA sequence. And I took DNA sequences from the A549 lung epithelial cells and predicted how likely it is that they would bind to the CTCF protein, which is responsible for how DNA is packaged within our cells. So the model I use is a variant or similar to the DeepMind architecture. And the reason why convolutional neural networks are so inherently powerful for genomics data is really simple. When you look at traditional image classification, it's two-dimensional, three-channel input, R, G, and B. But the way we look at genomics data is it's one-dimensional. It's 1 by M, where M is the length of the sequence. And it's a four-channel input for A, C, G, and T, the four nucleotides that are the building block of our human genome. And so we use essentially this very similar function as traditional image classification. We use convolutional filters, rectification layers, pooling layers, and fully connected neural networks. And what makes this method inherently powerful is through backprop, we can simultaneously update the thresholds, the weights, and the convolutional filters in real time to improve our prediction accuracy. And now I know that many of you are asking, why are we using genomic data with convolutional neural networks? Isn't it text? Wouldn't that be more of a recurrent type of application? But let me explain. When we look at traditional image data and how it's processed through convolutional filters, what we're doing is we're essentially calculating the dot notation for every single pixel value. So the filters, as you can see, rotate across an image. But the way that we can use genomic data in the same lens is we process it using one-hot encoding. And one-hot encoding allows us to take DNA sequences and process it into 4 different channels. For example, A is 1000 and C is 0100. And after we go through this process, we form a 4 by M matrix where M is the length of the number of nucleotides in our sequence. So it's almost analogous to an image. So essentially what we're doing is we're taking genomic data and we're making it into images that can be processed easily and understood by convolutional neural networks. And so the type of data that I'm using is ChIP-seq data, or chromatin immunoprecipitation data. And why it's so powerful is it allows us to quantify different binding sites in DNA sequences. When you look at this graph, you can see many different peaks. And what these peaks represent are different genomic regions within an entire gene that are hotspots for DNA protein binding, or where it's likely that a protein will attach to a DNA sequence and begin the process of transcription. So when we align this with sequence data, we can form target values 0 and 1 for whether at a specific location a DNA sequence binds to proteins. And so the machine learning pipeline we're using is we're inputting a set of sequences S separately into our machine learning model. And the output is essentially a binding score between 0 and 1. And we use the negative log likelihood objective to compute the loss function. And the reason why we use this is because our targets in this machine learning classification scenario are 0 and 1. If we were to have targets that were, say, between 0 and 1 and we wanted to calculate the difference, we would use something like mean squared error. And now I really want to focus on the convolutional stage. The convolutional stage is really inherently the strongest part of this architecture and why it works so well. So I use 64 different convolutional filters, which are either 4x4 or 3x3 convolutional kernels. And so we take in this one-hot encoded sequence, pass it through this convolutional filters, and it outputs us an array X. And the array X essentially follows the dimensions of n plus m minus 1 times d, where n is the length of the sequence, or how many nucleotides it is. M is the number of parameters in each convolutional filter, and d is the number of convolutional filters we're using. So when you try to visualize this array, what you're getting is by every single nucleotide in the entire sequence, we have the output of each of the scores for every single convolutional filter. So say 64 by n, where n is the length of the sequence. So after this, the variant of the DeepMind architecture that I use uses global max pooling instead of local average pooling. Because in the last couple of years since DeepMind, we've now found that max pooling is so much more powerful than average pooling. In addition to this, I use 2 fully connected neural network layers, and 2 output neurons for 1 and 0. And so where we can apply this towards genomics research is to predict the effect of different genetic variants. Let's say we take a regular non-mutated DNA sequence. We pass it through a pre-trained architecture and we get the binding affinity, a number between 0 and 1. Now, let's say that we found a mutation in real life. Let's say that this DNA sequence now has a mutation that causes it to go from C to G. We would input this same DNA sequence into the pre-trained network and compare the two different binding affinities. So let's say for the non-mutated version, we get a binding affinity of 0.89. But for the mutated DNA sequence, we get a binding affinity of 0.23. So here, because there's such a large gap, we can fundamentally understand that this has a drastic effect on DNA protein binding. And so from here, we can start to do further research to figure out whether this mutation is pathological or disease-causing. And so many of the diseases I talked about today, like Rett syndrome, are fundamentally can really be benefited by using this type of model on the genes that they are affected by. And so a really good visualization for this is mutation maps. And what mutation maps allow us to do is convey two different types of information. The first type of information is how strong the interaction is. So using how that number between 0 and 1, we can map out the height of a mutation. So if the length of the mutation, if the height of the mutation is incredibly high, we know that it's a strong interaction. Now the second type of information that these mutation maps convey is whether it's a positive or negative mutation. So as you can see here, the different red signals on the heat map signify that it's a positive mutation, whereas the blue signals on the heat map signify negative mutations. So from here we can draw a ton of different correlations. We can see that throughout the LDR1 gene, or cholesterol gene, Cs have a drastically negative impact on DNA protein binding. Because if you look throughout the gene, you see that C mutations have a blue signal throughout the mutation map. But on the other hand, we can see that T mutations have a positive impact on DNA protein binding. So perhaps a solution for a lack of protein production in the LDR1 gene could be creating T mutations in our gene that allow us to create more of the LDR1 protein. And all of what I talked to you about today isn't just theory. What really excites me about this research is that it's being put into practice. Just last month, Deep Genomics, a company run by Brendan Frey, one of the pioneers of this approach, announced the first ever drug candidate for Wilson's disease, a rare genetic disorder in children. And what's really crazy about this is that the entire drug was generated using AI. The target was found using a similar method that I proposed to you today, using AI, and the molecular characteristics of the drug were also generated using AI. In addition to this, recently Microsoft and Novartis entered into a $1 billion partnership to help accelerate AI drug design efforts. So their researchers are going to be working on how they can apply machine learning to diseases like macular degeneration. And why I'm really excited about this partnership and think that you guys should also be excited is that this work has the potential to rapidly scale up AI and drug design. So when you think about Wilson's disease, that's a disease that it's a rare disease, so it affects thousands of people. But when we think about diseases like cancer, where there's multiple different mutations in our genome that causes it, we need large-scale teams working on this problem. And for all of you in the machine learning space, this isn't a complex biological problem. It's a complex machine learning problem. And so I think all of us in this room really have the potential to have a huge impact on this industry and help make drugs cheaper and more accurate so that people across the world, not just in developed countries, but developing countries, can have access to medications for rare genetic disease. Thank you.